In genetics, epistasis is when the expression of alleles at one locus masks or modifies the expression of other alleles at another locus. Alleles are different versions of a gene, and loci are the specific locations of genes on a chromosome. Since one gene masks or changes the effects of another gene, multiple different genotypes lead to the same phenotype in epistasis. The genotype is the set of alleles that underlie a trait, and the phenotype is the physical characteristic of that trait. The fur pigment of an extremely rare species of giraffe found only deep in the African savanna is an example of epistasis. This gene is controlled by two loci. The first locus has the A allele, while the second locus has the B allele. We will see later that the gene with the A alleles is epistatic to the gene with the B alleles. There are three different phenotypes of giraffes found on the land. The first is the typical yellow giraffe, the second is the purple giraffe, and the third is the blue giraffe. If we cross two hybrid giraffes, we expect to get these genotypes in the offspring with a phenotypic ratio of 9:3 to 3:1. However, when two of these heterozygous giraffes mate, a different phenotype ratio results. When two yellow giraffes are crossed, we actually get 12 yellow giraffes, 3 blue giraffes, and finally one purple giraffe. These offspring show a 12:3:1 ratio. With this ratio, it can be assumed that single dominant epistasis is occurring. The following pathway explains how the dominant A allele masks the expression of the B allele. We start with yellow pigment, and an enzyme controlled by another gene on the chromosome can convert this yellow pigment to a purple intermediate pigment. However, the dominant A allele inhibits this enzyme, stopping it from fully functioning. If the inhibitor is not present, or in other words, the dominant A allele is not present, then the pigment will go from yellow to purple. Now, if a dominant B allele is present, it will code for a second enzyme that converts the purple pigment to the blue pigment. However, this can only occur if the A allele is recessive, since the first enzyme is needed to get to the purple pigment in the first place. If the A allele is recessive and the B allele is also recessive, the pigment will stay purple. Therefore, the only way the pigment can become blue is if there is a lowercase a allele and a capital B allele. Let's go back to our cross between two yellow giraffes, each with this genotype. The offspring have a phenotypic ratio of 12:3:1. Notice how all the giraffes with the capital A allele are yellow, regardless of the B alleles being dominant or recessive. Now let's cross a yellow giraffe with the genotype big A, big B, with a blue giraffe that has the genotype a, big B, little b. We have 8 yellow giraffes with the dominant A allele and 8 blue giraffes missing the dominant A allele but having the dominant B allele. None of the offspring were purple since they did not have both recessive alleles at each loci. The phenotypes show half yellow giraffes and half blue giraffes. In the case of this rare species of giraffe, the A locus, when it is dominant, masks the effects of the B locus, demonstrating single dominant epistasis.